Hello, I'm Barbara Konkle from the Washington Center for Bleeding Disorders, and I thank the organizers for allowing me to give you an update on the My Life, Our Future project. What I want to cover today is to briefly describe the value of knowing the genetics of hemophilia, to share a few results from the My Life, Our Future project, and to discuss how results may impact current and future care. So what's the value of knowing the genetic change, or we call it now the variant or mutation responsible for hemophilia in your family? So it's important that the knowledge can be used in really characterizing hemophilia in your family by testing family members to identify genetic carriers of the hemophilia gene, and importantly, to identify women and girls with hemophilia, to know who, for certain, who should have factor levels drawn to see if they also have hemophilia and would be at risk for bleeding, and also to aid in pregnancy planning, delivery, and early infant care. It also is important to help us understand who is at greater risk of developing an inhibitor to either factor VIII or factor IX once individuals are treated for their hemophilia. So in a broader sense, it's important to understand genetic influences in hemophilia. We know the impact on the factor genes, but likely how hemophilia is expressed, how it manifests in someone, is also determined by a number of other genes, which we can understand by studying genes outside of the factor genes, perhaps to understand differences in bleeding symptoms, outcomes such as differences in who develops more joint disease, how long the factor stays in the body, determine why inhibitors are more common in African Americans, and understand differences in bleeding between men and women with low factor levels, as well as other questions. So My Life, Our Future had two distinct components, one on an individual basis to determine the genotype and understand the impact of that on a person's hemophilia, and then bringing multiple samples and data together to really contribute to science and determining the future of hemophilia. So the structure was that the patient or parent authorized testing. The Hemophilia Treatment Center drew the sample and sent to Bloodworks Northwest. I direct the genetics lab there, and we were the core laboratory. Then we determined the genotype and sent a laboratory report back to the center. Over 80% of patients also consented to be part of the research repository, for which there were additional samples and data from the ATHEN data set. And then this can be used to address research questions, and the use is governed by a research review committee. Also importantly, all the samples and data in the repository people are identified by numbers. And so no one knows the identity of the individual, only the Hemophilia Treatment Center who initially consented the individual, and they actually can only relate it to the number if we ask them. So a few results. The project went from 2013 to 2017. There were over 11,000 individuals, 69% male and 31% female. Originally, it was focused on males and females with hemophilia and then expanded to known or potential carriers. About 80% hemophilia A, 20% hemophilia B, which is usually what we see, and about half were under the age of 20. We have just finished really compiling all the initial results and have submitted them for publication. In terms of race and ethnicity, about 18% were Hispanic or Latinx. In race, almost 1% American Indian or Alaskan Native, 3.5% Asian, almost 10% Black, about 0.5% Native Hawaiian or other Pacific Islander, 80% white. And when we compare this to the Hemophilia Treatment Center population, the percentages are almost identical. So what do we learn or confirm? Well, we've known that there are a lot of different genetic changes that can result in hemophilia, and I don't expect you to be able to read all of those shown here, but the different colors on the pie charts represent different types of changes in the hemophilia gene. The gray pie pieces in the severe hemophilia A are the inversions where there's a flipping of the gene back on itself, and that's the most common reason for severe hemophilia A. The big orange slices are missense. That's where just one nucleotide, one amino acid is changed, and that's most common in mild and moderate hemophilia A. And just to kind of understand what happens with these different changes, if we think for DNA, there are 3 nucleotides that then encode for an amino acid, which is the building block of the protein. And if we think of them as 3-letter words, then a missense, which we talked about, just changes 1 letter. And so it just changes 1 word or 1 code. And so that can have different impacts on the protein depending on what amino acid is changed. So it usually can do from mild to severe hemophilia depending on the change. Nonsense, there's a stop that causes it to quit making the protein. A frame shift, you can have an introduction of a new nucleotide or loss of one, and then it makes kind of garbly goop until it stops, and that often also results in severe hemophilia. Can delete a couple of nucleotides or insert them, or have large insertions or deletions, and the impact on the protein and the effect on hemophilia depends on the location and the size. So we did confirm there are many genetic changes. One of the things that was surprising to us, that throughout the project we saw new variants or mutations. And so shown on the right is a graph from that. And you can see as the red line, it increases for more novel variants as we tested more subjects. There was a little bit of falling off on the end, but it just shows that there can be multiple changes in the factor VIII or factor IX gene resulting in hemophilia. And we know that mutations are not race specific. So what did we learn or confirm about inhibitors? So it was known that they're more common in Black. There have been some controversy in terms of Hispanic patients, and we did show some effect of that in both hemophilia A and hemophilia B compared to white and Asian patients. We also found, and this was really given the size of our study, which was by far the largest genetic study in hemophilia ever done, that inhibitors appear to be more common in Native Hawaiian and Pacific Islanders compared to white and Asian patients with hemophilia A. The numbers are still small, and so we need more work in this area. And also because of the numbers, we could look at some of these very complex rearrangements in the gene, particularly factor VIII gene, and those individuals are more likely to develop inhibitors. So importantly, we have this large research repository, which is really a gift to the research community, which researchers then need to give back to the community with findings. And there's been now we're in our 2nd cycle of research applications. And so some of the questions being asked are, why do patients develop inhibitors? Why are Black patients more likely to develop inhibitors? Why are bleeding symptoms different in patients with the same factor level? I discussed there seems to be differences in bleeding between males and females, and there's a project to try to understand that, to understand why there's a difference in how long factor lasts, and there will continue to be more questions asked of this research repository. So with that, I'd like to thank you, particularly patients and families who participated in this project and the HTC providers who made it possible, as well as the core the American Thrombosis and Hemostasis Network, the National Hemophilia Foundation, and Bloodworks Northwest, who, along with funding from Biogen BioVeritiv, served as founders, and the research repository remains under the purview of Athen NHF and Bloodworks Northwest. Thank you.